At the end of the video overview of the immune system, we pointed out that the T helper and B cells in this image were likely memory cells, since the B cells were already secreting antibodies and the T helper cells were able to respond rapidly by sending signals. At the end of the handwritten tutorials video about VDJ recombination, you were left with the fact that there are now 1 billion circulating B cells, all with unique variable regions. These B cells and T helper cells are the most important parts of the adaptive immune system because they all have unique antigen receptors, allowing them to respond to virtually any antigen presented by a pathogen. We want to talk about how a naive B cell, which has never seen its antigen, becomes activated to make many copies of itself, which is called clonal expansion, and leads to production of antibodies that are secreted out of the cell to find epitopes on the pathogen. Unlike the innate system, which responds to the pathogen immediately, the activation of the adaptive system takes time because there has to be an interaction between the B cell and its cognate T helper cell. The innate system is limited, however, in the pathogens it can respond to. Remember, the TLRs in the innate system respond only to PAMPs, which are primarily bacterial pathogen molecules that are very different from human proteins. We need to also have a system that will respond to foreign proteins that might be similar to human proteins. The major pathogens with proteins similar to human proteins are viruses. Let's take a closer look at B cells. Here's one of the 1 billion circulating naive B cells. It has a nucleus and endoplasmic reticulum, as well as a BCR, or B cell receptor, which is the surface expression of the antibody with its unique binding site that will eventually be secreted by the clone of B cells. Additionally, this naive B cell has an MHC2 molecule on its surface, which has a variable, but not unique, binding site. To become activated, this naive B cell needs to encounter its cognate T helper cell that will bind to the MHC2 epitope complex presented by this B cell. So how does this happen? First, the B cell, through chance, encounters a viral coat protein it can bind to. It incorporates the virus in an endosome, breaks it down into small peptide pieces, and then as the MHC protein leaves the endoplasmic reticulum, the epitope is bound to the MHC2 molecule. This epitope MHC2 complex is then presented on the B cell surface. A T helper cell that recognizes a protein epitope from the degraded original virus is the cognate T helper cell. It is a cognate because it is recognizing a different protein epitope, but it is from the same virus. This binding of the cognate T helper cell activates the B cell, resulting in clonal expansion and antibody production. In summary, virus epitope recognition produces clonal expansion of B cells and eventually memory B cells. First, the B cell recognizes a virus coat protein epitope, then it encounters its cognate T helper cell that can recognize a degraded protein epitope from the same virus. The binding of the cognate T helper cell results in clonal expansion through T helper cell signaling molecules. Because all of this is occurring by chance, it takes time for the adaptive immune system's response. In this example, we have been referring to a virus as the pathogen, but the response would be the same to a bacterial pathogen. In the handwritten tutorials video, you learned about how the body makes unique B cell receptors and T cell receptors through VDJ recombination. This allows the body to respond to virtually any foreign epitope. There's another molecule that also needs to handle virtually any foreign epitope. That's the MHC molecule. There are two types of MHC molecules, MHC1 and MHC2. MHC1 molecules are found on the surface of almost all cells. MHC2 molecules, which we have just finished talking about, are found only on antigen-presenting cells, macrophages, dendritic cells, and B cells. MHC1 molecules, just like MHC2 molecules, present processed epitopes. If invaded by a pathogen, MHC1 molecules, which are on the surface of all nucleated cells, will present foreign epitopes. This signals cytotoxic T cells that the cell has been invaded and should be destroyed. Both MHC1 and MHC2 molecules are highly variable in their binding grooves, but not as variable as the binding groove on the B cell and T cell receptors. You can think of the binding groove as a bun and the epitopes that can fit in the groove as a hot dog. In contrast to the B cell and T cell receptors, which can only accommodate a single unique epitope, the MHC binding groove can accommodate many different epitopes. Because the MHC grooves accommodate multiple different epitopes, we only need a limited number of different MHC molecules. Humans inherit 6 MHC1 alleles and 6 MHC2 alleles, 3 of each type from each parent. In the human population, there are more different alleles for these 6 MHC1 and MHC2 molecules than for any other protein, on average over 100 alleles for each locus. It is this diversity of alleles, in combination with the ability of the MHC groove to bind many epitopes, that allows each individual to present the wide variety of pathogen epitopes. It's important to realize that during any infection, you are generating many different T helper cells, antibody-producing B cells, and cytotoxic T cells. This is in response to the many different viral epitopes that are presented initially by antigen-presenting cells and later by infected cells to the cytotoxic T cells. To make lots of different antibodies and cytotoxic T cells against a single invading virus requires MHC diversity. Without this diversity, the invading virus can escape detection and kill the organism. This image is showing a B cell with both MHC1 and MHC2 proteins on its surface. They are inherited as a group from chromosome 6 and are variable because of the many different alleles for these genes. Each MHC molecule has 2 subunits, each of which is coded for by a gene. These genes are inherited as a group, so each individual has a haplotype defined by their parents' haplotypes. Your haplotype is a set of 6 paired subunit genes that you inherited from your parents. The variety is a result of the multiple possible alleles that you could have inherited, rather than a result of recombination. So your MHC molecule variety stays the same throughout your life. In contrast, the VDJ recombination is happening over and over again in your bone marrow to make unique B and T cell receptors throughout your life. MHC molecules first became known in the scientific community when organ transplantation was just beginning in the 1960s. The variability of MHC molecules on the surface of cells resulted in the rejection of organ transplants between unrelated individuals. If you looked at unrelated humans, you would find the chance of having the same haplotype is less than 1 in 100,000. MHC molecules from an unrelated donor are recognized as foreign antigens by the recipient, just like a foreign pathogen. This is problematic from the perspective of transplant surgeons, but the evolution of a variety of MHC molecules was important for the survival of our species, since this was necessary to recognize the variety of pathogens we are exposed to. To check your understanding, go on to answer the questions that follow.